Okay. Welcome, everyone, to the Complex Causes of Autism webinar series hosted by Autism New Jersey and sponsored by the New Jersey Autism Center of Excellence. I'm Dr. Suzanne Buchanan, Autism New Jersey's Executive Director. This is day 3 of our 3-part series. For those of you who don't know us or just joining for the first time, Autism New Jersey is a nonprofit that helps individuals, families, and communities. Our helpline staff connect individuals and their caregivers to reliable resources for autism diagnosis, treatment, education, and more. We help government leaders create autism-friendly laws, and we inform healthcare providers, law enforcement officers, and other professionals about autism best practices. Through our awareness and engagement efforts, we work to create communities where people with autism are accepted and thrive. I have with me Cassidy Grom, our Communications Director. Cassidy will help facilitate the Q&A session during the second half of the webinar. Many of you submitted questions for our presenters when you registered, so we have some of those teed up. And if you have questions during the webinar, please submit them through the Q&A feature at the bottom of your screen. We'll try to address as many as we can, and please know that we are saving all of them. We're saving each and every question we're planning on how to get them answered in the future. Okay, just a few more housekeeping notes. We'll put a link to a certificate of attendance in the chat at the end of the session. Actually, correction, it won't be in the, yes, it will be in the chat. The questions go in the Q&A. Okay. And we will be making these sessions available on our website in the next few weeks. So you can go back and view ones, maybe to hear a lesson again, or share them with a friend. And also, some people have asked for the handouts. We're working with the presenters to see what we can share, but we expect we'll have some handouts available on that same webpage where the talks will be. So again, we are so thankful to the New Jersey Autism Center of Excellence for being such a great partner and sponsoring these talks. And last but not least, if you have any questions about autism resources, how to access services, or about our statewide initiatives, you can always call us at our 800-4-AUTISM helpline. So, next slide. We're here today to continue sharing what researchers know about the causes of autism and to help answer some of your questions. Unfortunately, I think we all know there's been a lot of misinformation about autism, so it's important that we learn about credible research from reliable resources. And we've had more than 1,400 people register to join us this week. It's an incredible testament to our community's desire to learn more about the scientific underpinnings of autism. In the first session, we learned about the potential genetic causes, and participants submitted more than 100 questions. During the second session, we learned about environmental causes, and participants submitted more than 110 questions. Today, we'll learn about brain development and structure, and we very much hope you keep the questions coming. We all have so much to learn. So, here today is someone who can teach us everything, hopefully everything we need to learn, and someone whose work I've admired for more than 20 years. I'm thrilled to introduce Dr. Manny DiCicco-Bloom. Dr. DiCicco-Bloom is a professor of both neuroscience and cell biology, as well as pediatrics at Rutgers Robert Wood Johnson Medical School. He studied at Princeton University, received his MD from Cornell University Medical College, and following postdoctoral studies in developmental neuroscience, trained in pediatrics and child neurology at New York Hospital Cornell Medical Center. His research seeks to understand the molecular and cellular pathways that regulate the production of neurons during brain development, something that I learned is called neurogenesis, and how genetic and environmental factors disrupt neurogenesis and contribute to neurodevelopmental disorders. Currently, he's exploring how the effects of antibiotics on the gut microbiome and brain development are significantly enhanced in mice that carry a human autism genetic risk variant. Dr. DiCicco-Bloom has long served scientific and advocacy organizations setting strategies and priorities for translational research at the NIH, the Federal Department of Defense, and the Society for Neuroscience. Also at disease organizations, including National Alliance for Autism Research, Autism Science Foundation, Autism Speaks, and the Autism Tissue Program, and is the scientific advisor of the Eagles Autism Foundation. Please join me in giving a warm welcome to Dr. Manny DiCicco-Bloom.
Thank you, Dr. Buchanan. Pleasure to be here. I want to thank Autism New Jersey and New Jersey Autism Center of Excellence for setting up these really important webinars at this point in time. And I know you've had the opportunity to hear about some of the complex factors that play roles in autism, including genetics from Dr. Manzini and environment from Dr. Halladay. And now I'm going to talk about how genes and environment impact brain development, mainly prenatally, while the infant is a fetus in utero. And what are the processes that are altered that contribute to autism spectrum and other neurodevelopmental disorders? Well, as a child neurologist in the clinic, I see neurodevelopmental disorders. And as you can see on this slide, there are quite a number. And importantly, these different disorders actually tend to overlap. And so individuals may have one or two or even three at the same time. And interestingly, since they all have to do with the brain development, many of the factors that contribute to one, let's say autism, may also contribute to another, like ADHD. So in the clinic, we frequently will see children with autism spectrum disorder or global developmental delay, intellectual disability or learning disabilities, attention deficit hyperactivity disorder, specific language impairment, or Tourette's syndrome. And so these are common things in the clinic, but also another developmental disorder that presents later in life includes schizophrenia and bipolar depression. And so, as many of you may be aware, let's discuss some of the symptoms in autism. Of course, one of the things that new parents might experience with a child who is on the spectrum is they may have troubles with eye contact. And this is kind of a hallmark feature, such as this child here. While you may be speaking to them, they may be looking off, or there may be a reduced response to name. Another issue can be following the gaze of an important other, like a parent looks to the side at something exciting, but the child doesn't follow their gaze or point, follow pointing, when this is referred to as joint attention. Overall, some children will have reduced facial expression or an inability to use facial expression to communicate or understand what expressions mean. In the other areas of the disorder, there are difficulties with excessive or repetitive behaviors or activities. So, for example, we can see the cars lined up here, and lining up toys as opposed to playing with them may be an issue. Well, there might be great attention to details like having a truck, but instead of rolling it around, sitting and spinning the wheel. So this is an activity which is atypical for age. Although many of the symptoms that we see in autism actually are part of normal development, but they may persist beyond their normal developmental period. At the larger level, there can be movements of the fingers or flapping of the arms or spinning of the body. And then there can be adherence to routines and changing the pattern of going to bed, like brushing the teeth or reading a story, can result in challenges. And finally, in the area of language, and when language is present, there can be unusual use of phrases, there could be memorization of songs but not understand language, or repetition of words that are heard, echolalia. And so decades ago, Bob Schultz, who runs the Center for Autism Research at CHOP, and Ami Klin, who runs the Marcus Center Atlanta, they were both at Yale Child Study Center, and they developed a kind of simple technology which helped us to quantify eye tracking. So if we look at typical participants who are looking at pictures, if we look at the left, if a typical individual will see a picture, they'll tend to look at the eyes and spend more time at the eyes, whereas an individual on the spectrum may not focus as much on the eyes, might look at the chin or the mouth or the ear, or actually somewhere else. And they could use little lights that actually shine on the eye so you can actually see where the eye gaze is. So this is a hallmark feature that kind of distinguishes this social disability in autism from other social challenges in other disorders like schizophrenia, for example. I think we've all heard the recent the report by the CDC of the prevalence of autism now being 1 in 31 individuals, about 3%. and about 40% of boys and a little less in girls have intellectual disability. And as we're well aware, males are more likely to have autism than females, although as we've had some discussions, part of the issue has been that the criteria for diagnosing social differences are actually going to be different in girls than in boys. And this has to do with the historical initial design of clinical tools basically focused on boys. So as a clinician, we actually make our diagnoses based on the Diagnostic and Statistical Manual, number 5, DSM, and I'm sure you're familiar with this. The two primary core problems include persistent problems with social communication and interaction that occur in multiple contexts, not just the classroom or not just at home, but throughout. And then the other is restricted and repetitive behaviors or interests or activities. These need to be present and will frequently are recognized in the first 2 years of life. They actually need to be recognized before the age of 3, but frequently are present before that. They have to be impairing other typical activities that are developmental stage appropriate. And they are importantly not better explained by intellectual disability or global delay. In addition to these two primary areas, we now also have specifiers, which are graded in severity from 1, 2 to 3. And I won't get into the details of that, but with or without intellectual disability, with or without language impairment, and then the presence or absence of identifiable genetic, medical, or environmental factors. Now, we did hear in our previous seminars of single genes that cause syndromes that have high prevalence of autism, and this includes Fragile X syndrome, tuberous sclerosis, and another one would be Rett syndrome. These are single gene monogenic disorders. But the other specifiers can include the presence of epilepsy, having an environmental exposure to an anticonvulsant, valproic acid, which is Depakane, which we now avoid using in females, and then also problems with birth weight, very low birth weight. But importantly, if you have autism, about 70% of the time, you're going to have another one of those developmental disorders, which I listed earlier. And up to 40% of individuals can have two. The most common associated abnormalities include ADHD and anxiety and mood disorders. By adolescence, about 30% of individuals on the spectrum will have epilepsy. And as we've heard about now, with the large amassing of more individuals in samples, intellectual disability occurs in about 40% of the time. So as we've already heard in our first two seminars, genes play a really important role, but they're not the only important factor. Environment plays a role as well. And as we heard from Dr. Manzini, twin studies, when you've got two individuals who have the same genetic material, monozygotic twins, the concordance, the occurrence of autism in one will predict autism in the other, not 100% of the time, but between 60 and 94%. So that means there are non-genetic factors. And of course, this is occurring during gestation. And you keep in mind that different individuals, different fetuses, will each have their own placentas. And so that can play a role. Second of all, if you've got fraternal twins, the concurrence of both of them having a diagnosis can vary from 10 to 17%. And then finally, as we know, if autism in an individual has to do with the genetics of the parents, then there's a 10 to 20% likelihood that the next child in that family will also have autism. So these all imply important roles for genes, but also indicate that not everything is genes, and then the rest is, we use non-genetic factors as environment. I've already referred to medications, valproic acid. We discussed yesterday the role of maternal infection, challenging deliveries, whether or not a child's very low birth weight or has difficulty coming through the birth canal, and differences now we are understanding in the placenta. of maternal stress or environmental chemicals. So let's just talk about the brain for a second, and then I'll talk to you about building the brain. I'm sure you're all familiar with the frontal cortex. That's what allowed you to plan to get to this webinar for me to prepare it. And this has everything to do with language and motor development. On the opposite, and of course, the frontal lobe here is right here at the forehead. And this is the nose, and this is where the ears would be. So this is the whole brain. This is the cerebellum and brain stem. And this is the spinal cord, which has been pulled up to give you this picture. So in the front, we plan and we talk and think. In the back, we can see this is the occipital or visual cortex. Temporal lobe is here. This is really important for understanding language. It's the location of the hippocampus, which is really important in learning and memory. And importantly, in front of the hippocampus is the amygdala, terribly important for social and emotional and fear. And what's important is that if we get information about hearing a bird, for example, and seeing a bird, these two primary areas have to be brought together. And that occurs here up in the parietal lobe, the association cortex. Now, below the top of the brain, down below, basically behind your ears and lower, is the cerebellum and the brainstem. Each of these regions have been found in different individuals on the spectrum to have changes that occur development before birth. And so I'll go into what it takes to make nerve cells for here, and I'll describe some of the changes that we see in the brain. So before we go there, let's talk about nerve cells. We heard from Dr. Manzini that we have 80 billion nerve cells. And so hopefully a little bit behind me, and right here is this beautiful green cell. This is a single nerve cell. All of the nerves, this is a culture dish. We cultured some cortex neurons from embryos, in this case from a rat, and they're all red. And they're red because they have the cytoskeleton, the skeleton molecules of nerves. Well, this particular nerve right here, we did a genetic manipulation, the kind of manipulation we now are considering doing in individuals who have single gene disorders. I know Dr. Manzini discussed this. So this nerve cell is green because he put the green fluorescent protein from the jellyfish in it, so I can look at it. And if you have a green signal and a cytoplasmic signal, green plus red in light ends up being yellow. And so that's how we know this genetically altered nerve cell is in fact a typical cortical neuron. Well, let's go to a diagram and talk about the parts. So here is that cell body, and in the center is a nucleus where we know that's where the chromosomes and the genes are. And this big cell body puts out a very long process extension, as you saw in the picture. This is called the axon. And the axon has branches, and we call these axon terminals. Off of the cell body are these kind of hairy things. We call these dendrites. So we've got dendrites and axons coming off of neurons, and these are called neuronal processes. And sometimes I'll use the word neurite. Well, when a nerve cell makes a synapse, a connection to the next nerve cell, we call this connection of the axon terminal here, which has neurotransmitters like glutamate and GABA ready to be released when action potentials travel down the nerve to cause release of the transmitter. And so then that is on the dendrite of the next cell here, which then has receptors, which allows the glutamate here to signal to the next cell. And so what we know is that all of these different parts of the brain can be altered in some individuals with autism. And so now let's talk about brain growth. As a child neurologist, I know the brain starts really small, right here, 25 days, and this is how big it will be after 9 months. And at that point, the nervous system is just a tube. And at about 40 days, this tube starts to develop 3 regions: the forebrain, the midbrain, and the hindbrain. And this is due to proliferation of neural stem cells, making more neurons. And you can see that over time, different regions grow. The cortex becomes folded, so there's a lot more place to have nerve cells make connections and be influenced by learning. Well, so this is a picture here of a 40-day human embryo. Here's the forebrain and the eye, and here's the midbrain, and this is where the hindbrain will be. And this is the spinal cord, and here's the arm and the leg and the heart. Well, if we cut across that here in the forebrain, just basically like this on my head, this is what you'll see in an embryo. And this picture actually comes from a mouse, but this kind of a picture looks similar from a mouse, a rat, a monkey, and a human. And in fact, what's important, and the reason we use models, is we can't do experiments on people. And so by having convenient model systems, and for example, mice, we can study the processes that give rise to the brain. And even though a mouse brain is 1,000th the size of a human brain, many of the genes that give rise to the mouse brain are also present in humans. And so we can ask about how genes work to change building the brain and ultimately how it might impact behavior in a mouse, which to some degree can reflect the circuits in the brain that give rise to those behaviors. And so in this cross section, you'll see here, this is fluid-filled space. We call these ventricles. This is cerebral spinal fluid. And this green region here, which has this transcription factor PAX6, these are the cells that are proliferating. When these cells stop proliferating, they turn off that gene and turn on a red gene, TBR1, and these are postmitotic neurons. So they've migrated from the proliferative region to what will be the cortical plate. And so what happens during this process? So I actually described it to you, but let me show you a picture. This is a slice of the ventricular zone and embryonic cortex. And it's actually being a movie. This is time 0, and this is 20 hours, 36 hours, and 114 hours, looking at the same cell having that GFP as I showed you earlier. And here, this cell is dividing in 20 hours from one cell to cut in half, undergoes mitosis, and gives rise to two cells, one that's got a white and the other one has red. Well, what happens over time? Well, we can see that the white cell is actually migrating away, and it's actually being guided to some degree by this process. Whereas the other cell, which divided as well, there were pairs, becomes a stem cell and proliferates and gives rise to two new cells. And so this is the process of neurogenesis, generating neurons. And so what we know about the genes, many of the genes that we refer to that are important in autism, is they are actually expressed at this time when these processes are going on. And so it's logical to ask about, when we find genes that might be associated with autism, do they affect proliferation? Do they affect cell migration? Do they affect the growth of these processes, neurites or neuronal processes? And so how can we do that? Well, we can't do it in people, but we can look at the brain of individuals who've passed away. We can look at post-mortem brains and ask about how they are built. There is no consistent abnormality in a brain. We can't do a scan and say, oh, that's autism. Now, that's different than our ability to look for example, Parkinson's disease and some signs of dementia. So if we look at a brain scan, we can't say that's autism or ADHD. But when you look at, this is the data, the description here actually relies on about 300 published brains compared to thousands of brains analyzed with Alzheimer's. What we see are about 20% of individuals on the spectrum have a bigger brain, macrocephaly, whereas about 10% of individuals have microcephaly, smaller brains. And to some degree, the numbers of cells is going to be dependent upon how much proliferation. So if a gene alters a proliferation schedule or the amount, we could give rise to smaller or bigger brains. But as I indicated to you, sometimes neurons don't migrate correctly, so they can be in the wrong position, or they could be in the wrong layer or organized differently. And so this is another process that's affected by genes and environment. And finally, and we heard some discussion about too many and too few, too much and too little of a gene can give rise to too many or too few neurons, but also to neuronal processes, having too many or too few processes, which give rise to synapses. And I'll show you a picture of that in a moment. In addition to changing the numbers of synapses, we could have no change in that, but the transmitter system might not be normally responsive, or when we measure the electrical activity, it may not be working right. So even if the neurons are there and the synapse is there, their connection, their activity, their function may be altered. And again, all of this is happening before birth. So you've already seen this picture, and I'm just bringing you back up, the cell body and the nucleus, the axon making a synapse on the next cell. Well, what do we know about these synapses? The synapses on a dendrite actually look like this. Here's a cartoon. Here is a dendrite on that cell body, and here's that axon terminal with the transmitters. And this has a mushroom shape. In a typical brain, here's an example of a mushroom shape. Here's another example of a mushroom, and here, well, it's a little out of focus. Well, you heard about Fragile X syndrome. It accounts for about 1% of autism, and up to 40% of individuals with Fragile X will be on the spectrum. Well, what they found in individuals with Fragile X syndrome and mice that are a model is that there are too many dendritic spines. And instead of having a mushroom shape, they will look like a long stalk. Look at that. Here and here and here. So having too many synapses actually doesn't make you smart. It can give you problems. Well, so this is an example of a monogenic disorder where disturbing the biochemistry and the cytoskeleton gives rise to too many synapses. Are there other examples of this? Yes, another single gene monogenic disorder, tuberous sclerosis, gives rise to too many dendritic spines. So in the clinic, when somebody comes in for developmental issues, I will look at a person, and on some individuals, those with tuberous sclerosis, some of them will have these bumps on their face. Their medical name is angiofibromas, and they could be elsewhere on the body. The other hallmark of this is changes in pigmentation. This is the back of the legs. And you can see instead of having typical pigment here, there are patches that have reduced melanin. Well, importantly, in the embryo, there are three major layers, and the outer layer, called the epidermis, gives rise to both the skin and the brain. And so if we see changes in pigmentation on the skin, we may see, as pointed out by these arrows, changes in the nerve cells in the brain. We call these tubers. And so a person with tuberous sclerosis has these malformations, these nerve cells which are not differentiating normally. They also can have these nodules along the ventricle, which either are full of fluid or can be growths. What do we know about the nerve cells that are made in tuberous sclerosis? Well, let's again look at those dendritic spines. In a neurotypical individual, an 8-year-old neurotypical child who passed away, unfortunately, from a disorder, you can see the dendritic spines here. These are typical spines. But as we get older, individuals, here's an 18-year-old, you can see that the numbers of spines diminish. Well, why is that? Well, we know that during early childhood of rapid brain growth and so much learning, we exuberantly make tons and tons of dendritic spines and synapses. And then as we mature, we prune them away, we reduce the number because we make them more specific and get rid of the excess ones. However, in autism and in tuberous sclerosis specifically, even though there might be normal numbers of dendritic spines, like a typical 8-year-old, when they get older in their adolescence, those don't go away. So two monogenic disorders, Fragile X syndrome, tuberous sclerosis, too many dendritic spines, too many synapses, and interestingly, they have a high prevalence of seizures, which might occur with excess dendritic activity and synaptic function. Well, what's responsible for this pruning? A cell called microglia. So during development, from fetus to adulthood, we reduce the numbers of nerve cells total, excuse me, and the numbers of synapses. And in part, this is regulated by a special cell in the brain called a microglia. This is a picture of microglia in a rat brain in the embryo, in newborns, and adult. And the blue is a nucleus. And these processes here are important for them to feel out the environment. And the microglia can be considered the gardeners of the brain. They do the pruning. And so here's an example of a monkey brain, a rat brain, and the human embryo at 17 weeks. And in this case, the nerve cell nucleus is pink. And what we can see here is that this microglia is basically swallowed this nerve cell. And here we see that in a rat. And here in a human, what we know is that when green and red overlap, they turn yellow. So these are the normal cells that prune the numbers of neurons and synapses. And if they don't work correctly, we could have too many or too few neurons or synapses. So we've talked about the building block to the brain: proliferation, migration, process outgrowth, making synapses. Well, what does this have to do with the function of the brain, the electrical signaling, the connections and function? Well, one way to look at that is to do functional MRI scans. And so this is quite an old study, over 20 years. And I'm going to compare what a brain looks like. This is an MRI. Again, it's a section like this on the brain. And so here is a left cerebral cortex and right cerebral cortex and the cerebellum. And if I put my finger on my nose and then go to my finger and go back and forth, my finger stops, it accelerates, it moves constantly, then it slows down. This coordination of knowing my sensory, where I am in space, and modulating my motor activity is what we thought the cerebellum always did. And if we look at a typical brain, the cerebellum is active during motor tasks, and that also occurs in individuals on the spectrum. But as I'm speaking, if somebody calls your name, you're going to change your attention from me to that person. That's changing auditory attention, auditory networks. There's no motor movement involved there. But what we see in the typical brain is we activate the cortical hemispheres, but also the cerebellum. Whereas an individual on the spectrum may have no or little activation during changing attention. Does that have something to do with not responding to your name? So the brain can be structurally typical on an MRI, and I've said this before, but its function, its connections, or how it uses those neurons may be altered. So this has to do with the function of the brain, the connectivity. And so here is a picture of a 26-year-old male, typical or autistic. And these are a picture of all these axons, all these connections from the front to the parietal cortex to the occipital. And here's an autistic individual, and you can see that they might be aggregated and areas that are missing. So this is one way axons and dendrites could be different. The fibers may not spread or connect correctly. Well, what about the connections between neurons and brain regions? I think you may have heard in the news that we talk about maybe there's less connections. I just showed you a picture of the brain where functional MRI showed less connections between different regions. Well, we've actually now, and many studies have been done, thousands of MRIs, and we can talk about reduced connections, hypoconnections, or hyperconnections. This is a cartoon, which I think helps us think about this. Here's the front, the planning cortex and the visual cortex. In some individuals, there actually might be increased connections within an area. I might be an artist with increased connections in my occipital cortex. I might be a really smart scientist with increased connections in my frontal cortex. But some individuals, and so this hyperconnectivity might actually have something to do with savant abilities that we hear about. For example, memorizing all the addresses in the phone book. But do we know what to do with those addresses? And so there might be failures of specialized information connecting to different lobes. And so maybe there's a long distance reduction in connection. And so even though different areas can see or hear, maybe there'll be delays in processing that. So maybe reduced connections long distance wise underlies the delays in social and communication abilities. Well, so how can we think about this information of connection? Can we use it to look at people to understand whether they have autism? Well, there has been some recent progress in this, work by Joe Piven and Heather Hazlitt at the University of North Carolina and many others, and they've looked at baby brains. And I mentioned to you, if you have a family with one child with autism, the next child can be followed, the baby sibling, because they'll have a 10 to 20% risk of developing autism. And so what they found by doing these MRIs of the structure and the function of the brain is they can look at the brain at 6 and 12 months. And based on knowing about the structures of the brains and the connections, they can accurately predict about 80% of the time whether that child in the first year of life is going to have autism by 2 or 3. And so this is just an example. This is the top of the brain. This is the front and the back. The blue areas are pictures, regions of the brain, which are actually different at 6 months. And this is just different views of the brain. And the red areas are regions that are different at 12 months. And based on basically AI learning models, they can now predict, if they take pictures at 6 and 12 months, whether or not a child in the first years of life will develop autism. Similarly, looking at 6 months only, if they look at increased connection, indicated here by blue, or decreased by red, that single picture looking for connections can predict autism again about 80% of the time. So what's this got to do with genetics? Do genes affect connectivity? Can we use knowledge of genes and connections to make predictions about people's function? Well, I took this slide from Dr. Manzini's talk, which was developed and provided by Alycia Halladay and the Autism Science Foundation, just to remind us that we know maybe about 30% of the genes that contribute to autism, but more than 70% we don't know. As a scientist, I can't get enough people with any single genetic disorder. But by using mice, by creating a mouse model, we can manipulate genes by removing them and asking what are the consequences for building the brain and for connections. And so, and within here, as you know, is Fragile X and tuberous sclerosis. And there are also copy number variations, of which I'll talk in a moment. So here's a picture, fortunately, that neither Dr. Halladay or Manzini showed you, but about 5 years ago, a large collection of those 30% of genes that are known. These are about 100 of them, which are really, really, many of them are rare, but they're really penetrant. If you have the change in this gene, you're high likelihood of having the disorder in certain families. And as you've heard before, many of these genes regulate expression of genes during development, when we're making neurons. They have to do with the synapses, neuronal communication, and they have to do with the cytoskeleton, which underlies neurites, axons, and dendrites. And so what scientists have done is they've used mice, as I indicated, as a model. We can't manipulate genes in people and ask what happens. But fortunately, we can make mice with genes that reduce connections, indicated by blue signal, or increase connections, indicated by red signal. So let me orient you. This is a mouse brain. This is the frontal cortex region. This is the cerebellum. And this is the top, left and right hemispheres. These specific gene areas, if you look at these different genes, we've studied in GRAIL2 and 16p11.2, but here's oxytocin receptor and SHANK3 and others. All of these mice have reductions in connections in the areas that are blue, whereas all of these mice have increases in connections in areas that are red. And here's Fragile X syndrome, tuberous sclerosis. Remember, we already saw there are more synapses, more dendritic spines, and their connectivity is increased. So can we use all these models and put them all together and talk about regions that, regardless of the gene, cause problems with connections in areas important for behaviors? Well, preliminary evidence suggests we can. So the frontal cortex and the nucleus accumbens, which is our reward center, these regions are really important for social interactions. And so of all these animals, these regions, the frontal cortex and nucleus accumbens, have reduced connectivity. And maybe that's why they're reduced social function. And also in this area, the motor cortex, there's reduction. This may be why there's some changes in behavior. Well, What does this stuff with mice got to do with people? Well, if you take a picture of a person, as I showed you a few slides ago, and you see reduced or increased connectivity, if it occurs in the motor region, you might be able to then wonder whether the genes that gave rise to this picture in a mouse might underlie the human disorders. And so by having these mice where we can ask about genes and their impact on connections, we may be able to relate them to human beings. So finally, let me close on a few mouse studies, just to give you a sense of how, as a scientist, I might ask how changing a gene impacts a social behavior in a mouse. Now, this is pretty funny. Here's a mouse, and it's in this box, and it can go through this tunnel to another mouse here, or a cup with no mouse. Now, a typical mouse wants to be with a mouse and not an empty cup. But if you had a social defect, you might actually spend as much time with a cup as with a mouse. We studied the Ingrail2 mutant, which is a common variant that contributes a little bit to autism, and we found that that mouse didn't really care to be with a mouse. But let's see what that looks like. Well, we're going to try one more time.
Manny, I think you have to switch back to your pointer,
Ah,
your mouse cursor, in order to click on it.
pointer here. Where's the,
Yep, you have it.
I have it.
Yep.
I'm not seeing pointer. Well, anyway, let's move on. So this mouse spends a lot of time over here. And so that is how we look at social behavior. It's one of the ways. Well, what about another kind of behavior, another way of looking at social behavior? Another way to look at social behavior is to look at a related rodent called a vole. And here are the There are two types of voles out in the prairie. There are very social voles, monogamous. They're social. They live together in burrows. Both parents take care of the kids. They stay together for a long time. And when the kids are crying, they get upset. And then there are kind of the stud voles, the montane voles. They're not aggressive to intruders. They like to live alone. They don't care of the kids. They have a lot of partners. Well, many years ago, Larry Young and Tom Insel at NIMH, they asked, why are these voles different? Well, they knew about a peptide, which I'm sure you've all heard about, oxytocin. It's a small protein that regulates social behavior. This is really important in pair bonding between an infant and a mother, but also in males and females. And as you know, you can go on the web and order oxytocin and give it to your partner to see if you can increase their sociability. Well, in fact, what Larry and Tom figured out is that the montane vole had very low oxytocin signaling. And when they manipulated genetically the montane vole, they could make them social like a prairie vole. So they established in this mouse that oxytocin was a real determinative. And because of that, in fact, there was many studies looking at oxytocin in individuals with autism. And after many studies, and then finally a very large study run at Columbia by Jeremy Veenstra Vanderweel and multiple colleagues, they did the human clinical trials, but sadly they failed for the whole group. But importantly, and we heard this earlier from Dr. Manzini and Halladay, there are likely many autism subgroups. Different genes give rise to common outcomes. And in fact, what we do know is that there are subgroups of individuals on the spectrum who will and do benefit from oxytocin. So finally, let me show you what we did to look at autism neural precursor cells in kids with autism in New Jersey. We obtained a whole bunch of families, and I'll only show you 3 of them. And in this case, what we're dealing with is a family with a boy with autism here, 3 different families, and those individuals have siblings, male siblings, who don't have autism. And so we took their blood and we made neural precursor cells, just like the cells we were talking about in mice. And we also have copy number variant 16p cells as well. But I'll just talk about these. But the biology I'm going to show you now is exactly what you'd expect me to talk about. And that is, I'm going to talk about neurogenesis. And so again, I'm going to look at proliferation, cell migration, and neurite outgrowth, because this is when and where autism-related genes are expressed. So what do we see in neural precursor cells made from kids with autism? Well, I think you might be able to see this on your own. We took neural precursor cells, stem cells, and put them in culture for 2, 4, and 6 days, and we watched in the sibling, there were more proliferation, more cells. And in their autistic brother, there was less proliferation. So differences in proliferation, if there are less cells, there might be microcephaly or not the right kinds or types of nerve cells as are needed. What about migration and process growth? Well, let's just look at the pictures. Here's a typical sibling. The black arrows point to a nerve cell body with a process. You can see here, 3 cells or processes. And statistically, the kid with autism in the family had less. And that occurred, these are the 3 families. You'll see that the percent of cells with neurites in number 1, number 2, and number 3 is always less than their typical neurotypical. So neuronal process growth is also affected. And then finally, if we put a ball of cells down and we talk about this ball is very bright, but around it is a carpet of nerves, precursor cells. And you can see that the growth, the migration of the sibling is greater than in autism. And that occurred again in 3 genetically different, idiopathic, undefined individuals. And so what I've said is that by looking at individuals with idiopathic autism or copy number variant 16p, the processes that occur during early embryonic development between 8 and 24 weeks in a human of migration, proliferation, and process outgrowth are fundamentally disturbed. Well, to wrap up from Dr. Manzini and Dr. Halladay, I talked about the developmental processes. And as you know, we've emphasized that having genes alone may not be enough. You may need combinations. Or you might have a genetic predisposition, which then makes you more sensitive to an environmental factor. Well, interestingly, as you know, the brain is very sensitive to environmental factors, prenatally and postnatally, and so is the gut microbiome. And so here's an infant, and here's the gut microbiome with bacteria and viruses. And as we know, about 80% of all the immune cells in the body are around the gut. So what goes on in the gut and the biochemicals that are made there could be impacting immune cell development, endocrine hormones. They could directly signal the vagus nerve, which goes to the brainstem since it innervates the gut, and they can make metabolites, biochemicals. But as we also know, is that children get a lot of antibiotics. Typically in the US, the CDC indicates on average, we get 2 to 3 courses of antibiotics. Well, we give them for infections to make bacteria go away in our respiratory system, but they also reduce the antibiotic, the bacteria in the gut. And so a question arises, could changing the bacteria in the gut or elsewhere in the body impact brain development during this sensitive period? So Courtney McDermott, who got her PhD here after 5 years in the lab, is now doing her postdoc at Columbia, did this very experiment in collaboration with Dr. Martin Blaser, who's a microbiome expert here at Rutgers. And so what we did is we created a model. We took a mouse and gave them an early antibiotic, cefdinir. You may have given it to your child for an ear infection, a small dose, a typical dose for a brief period of time. And we gave it to mice that were in a family where half of them were neurotypical and half of them had the 16p copy number variant. And what we found is that, as we might have expected, antibiotics did change the microbiome and the biochemistry of the blood. It changed brain development in all the animals who received it, and it changed some of the behaviors. So changing the microbiome during development in a mouse can change a variety of things. But for those mice that had the genetic risk factor, their outcomes were worse. including the numbers of neurons in the brain and the kinds of behaviors they manifested. So this is an example how by knowing about genes and knowing about environment, we then ask the question in a controlled experimental model to ask whether or not this association might actually play a causal role. Keep in mind, as Dr. Halladay said, associations can be raise a question, but they don't tell us about causality. Here in this mouse, this is causal, but that doesn't mean it actually is causal in people. That will require a lot more study. And so I'll stop there and open up for questions.
Thank you so much, Doctor. Oh, let me turn my camera on here. And thank you to everyone who's been attending over the past week. I think that right now, in this day and age, there's a lot of misinformation or confusing information. So on behalf of Autism New Jersey and Executive Director Suzanne Buchanan and myself, we just want to thank you guys for coming and for seeking out scientific evidence and factual information. So we know that, this was 3 hours of your week that you could have spent doing something else. So we feel really honored that you chose to stay here with us. We will have a few minutes for question and answers, but before we get to that, I just wanted to give you guys a few quick reminders. You can always call our 800-4-Autism helpline to ask about things like a diagnosis or treatment options or schooling options. Our expert helpline staff is really there for you from birth all the way through adulthood. So feel free to call at any time. If you came to us through a different organization, if you saw it on social media or heard about it from a friend, please don't forget that you can sign up for our e-newsletter as well, where you'll get to hear about more of these sorts of webinars and resources. So if you go to autismnj.org and scroll down, there's a big orange section at the bottom of our website where you can subscribe to our newsletter. And last, I just wanted to remind you as well that some of you guys needed a certificate of attendance for attending, and we did put that in the chat earlier. So you can download it, you can click the link. It seems like our colleague Jeff provided multiple ways for you guys to access that. So please feel free to download that. Oh, sorry. And one more reminder, we are going to send out a survey later this evening asking you guys how you felt about the webinars, what we could do better, what we did, really well. It should only take about 3 minutes to fill out. So if you see that come into your inbox, please fill out our survey because we want to innovate and serve the community as best as we can. So, Doctor, are you ready for a few questions?
Absolutely.
All right. So we had a lot of questions about kind of that early brain development. And so we did have a question, and I think a lot of parents have this question. It says, is there any explanation for children who are developing typically, but then have regression around the age of 2?
So this, of course, has been a question that's been around quite a while, and this observation has been made. There are several issues I think we need to consider. First of all, we have a very narrow, very specific explanation in 2 cases of genetic disorders. But that's not the generalized answer, but why don't I talk about those first? So there is a syndrome called Rett syndrome. It occurs only in girls who are born. It's such a problem for brain development that boys never get born. And these girls have a typical growth for a year to a year and a half. And then suddenly activities that they were able to do, looking at you, using words, using their hands to do all sorts of activities, start to stop. They plateau. They don't continue to improve. And then over time, they actually lose some abilities. They may stop looking. They may, instead of using their hands to eat or draw with a crayon or throw things, they may start to do repetitive movements. They may develop seizures, and they actually may lose motor control. So there are various levels at which, at a certain point following normal development, when the gene that underlies Rett syndrome, MECP2, is needed for the next stage of maturation, it all gets messed up because it doesn't get expressed. And so this Rett syndrome, you can look that up at the CDC or NIH, and you'll be able to see descriptions. Another example, and I think this was reviewed earlier this week, is that a very specific gene that has to do with neurons, the sodium channel gene, called SCN1A, this can interfere with electrical activity of the brain. And so a child who can develop typically for a while, when there is stress in their body, including getting infections or high fevers, they can then, because they're more sensitive to the electrical balance, start to develop seizures. And so this can give rise to a seizure syndrome, which then continues. Keep in mind that fever is an accompaniment of getting an infection from other family members, and also the fevers that occur with vaccinations. And so these are two genetic causes that we know predispose to a problem. But more generally, since the concerns about early developmental, what the term has been used, regression, a number of studies have been performed, particularly in families with their first child, where they're learning to be a parent, as I think many of us know. It takes time before you actually realize there might be a difference. Well, if you go back and look at movies of infants in families where everything was fine and then at 2, they went backwards. If we go back, and multiple teams in multiple countries have done this, if you look at home movies, we actually see as clinicians that the challenges, the differences actually emerge earlier. So it isn't that there's completely typical development and then suddenly an event occurred, although again, that can be the case. It's that it takes time for those around to identify.
Thank you. Yeah. And kind of as a follow-up to that question, we had several questions asking, do factors prenatally when the baby, is still with its mother, are those more likely to cause autism, or is it after the baby's born? It sounds like from your presentation and from the scientists that we've heard previously that a lot of this occurs prenatally before the baby is born. Is that correct?
That is correct. As all of us have emphasized, the genes and the factors are impacting the brain development. And keep in mind, although the samples are small, if we look at the brains of individuals, we see problems of the proliferation, the growth, and connections that only occur when before the baby is born. But keep in mind that we don't know there's a problem with an area, even if it's built wrong, until it's engaged. I do finger to nose, but think about if those of you who have children, when they're actually able to do finger to nose. It takes time before they can do that. And so we wouldn't know there's a motor problem until we call on it. And we see this with children with ADHD or learning disability. Until the demands of their typical growth are present, we may not see those challenges.
And because it's come up so frequently in our questions, I think people still are wondering about the rhetoric around vaccines causing autism. And for a lot of these parents, they get vaccines around the age of 2. And so that's also when they're noticing differences. What is your comment on that cause of autism?
Well, I think as Dr. Manzini and Dr. Halladay and many other leaders of the field, probably 40 studies have been done around the world, multiple countries, not only the US, but multiple actually countries where they have a birth registry where every infant is known, like in Norway and in Sweden and in Denmark. They know the history of every child. And these studies have demonstrated that there is no increased prevalence associated with vaccines. I will say, sadly, about a decade ago, because families did not want to vaccinate their children in the state of California, they actually set up an experiment we ethically could not do. And then they looked at children who weren't vaccinated versus those who were. And sadly, there was more autism in the families where vaccinations were not given. And this has to do with the fact that infections in the mother, infections in the child are a challenge to the developing brain. And so that can be a negative factor. And keep in mind the other issue about vaccinations, one of the questions was whether the preservative caused a problem, ethyl mercury. But ethyl mercury was removed decades ago, and autism has not reduced in its prevalence. It continues to rise. And as I think we discussed before, the increased prevalence primarily has to do with us clinicians learning more about how to make the diagnosis, with us identifying girls better than we did in the past, and the fact that the criteria for making the diagnosis have changed. And finally, because now healthcare is more generalized, and so many in cultures and groups of individuals who weren't getting good healthcare now are. And the recent increase in prevalence in autism occurs in Black and brown families who didn't have the option of getting care or had other stressors in their life that prevented them from getting diagnoses. So when you look at now all types of individuals in the US, the prevalence is the same. And I think there was a question earlier about whether the prevalence in the US is similar or different than the rest of the world. And the answer is they're all very similar.
Thank you for that. That was very insightful. We're going to do a quick speed round if you're okay with that. So
Absolutely. Let me get a touch
we'll
of
try to
coffee.
get through
Go
as
for
many
it.
questions as we can here. Yeah. So is the use of technology at a young age impacting brain growth? We had a lot of questions about iPads, using lots of screens, that sort of thing.
It's not the kind of research I do. I do think, as we've all seen, our behaviors as humans, as we get attached to screens and expectations and fulfillment, that does play a role in building the brain. There's no question. But I don't think there's any evidence, and I'm not really the person that knows this literature, whether there's any evidence that early screen per se. Keep in mind, there were concerns when cartoons were introduced in society, when television, I actually remember the first televisions. So everybody born before that didn't have television. So there's no question that using different capacities of human beings will develop different brain capacities as we grow. But I don't think there's any evidence to support that. But I would refer you to others who might know better.
Gotcha. Thank you. And then there was a question, is there a definitive test? Like there's a COVID test or a test for diabetes that you can get done to diagnose autism?
No, there isn't. No, there is no definitive test. And although we heard from Dr. Manzini about genetic risk, and there are organizations that are suggesting we can look for contributory genes, but that talks about the possibility, the risk that it will happen, but it doesn't predict the outcome. And so it's really important not to take the possible risk as a suggestion that it's present. And whether these are tests looking at genes, whether they're tests looking at antibodies in the blood or biochemicals in your blood, we are not in a position to predict.
Yeah, and from what I've heard, autism seems to be diagnosed kind of on that checklist, right, of social interactions and other things that you're basically just observing, right, as a clinician.
They are observing. Now, obviously I went over in a family with one child with autism. I talked about brain imaging and functional studies on the MRI. That helps make predictions of whether a child from a family at risk might develop autism, but we can't even say for those children, only 80% predictive reliability, 20% were equally at risk. So we can't do that.
Thank you for that. And then, of course, we have a lot of parents wondering, okay, my child got diagnosed, now what? And we had a question about how moldable is a child's brain and what can a parent do during those early years to help a child's brain develop?
Well, the first thing I do when I see a child with autism is I refer them to Autism New Jersey,
Well, thank you.
because as families, as clinicians, as educators, you provide such a broad base and deep experience in finding the best tools for diagnosis, of finding the kind of services. A child neurologist will refer to you or a psychologist or a developmental pediatrician, because there are multiple resources which have to do with behavioral interventions. Certainly loving your child, paying attention. A lot of the interventions that are being done with families have much to do with acquainting families with what the developmental milestones are. Even though a child may be smiling as you speak to them, that smile doesn't mean they understand your words. And those of you who have young kids, you'll also know that kids can say really remarkable things, but they're repeating what they've heard, but they don't understand the meaning. And so it's really important for families to be sensitive to different developmental stages. And as I said, the symptoms that were on that list are typical phases of development. In autism, they just may not be going away as predicted. And so none of these things are absolutely aberrant. They're part of development. So loving your child, seeking out guidance from your pediatrician or a developmental pediatrician or a psychologist, these are the ways that you can improve their growth by interacting with them.
Thanks, doctor. Yeah, and from my understanding, the ABA therapy, the applied behavior analysis, I think I have that right, is kind
Yes.
of the gold standard for therapy, and that can really help your child develop some skills.
Absolutely. And there are, as children age, there are different types of approaches. ABA is certainly important, as is parent training. But as they get older, sometimes there's social training and other activities that help an individual adapt. But understand that different families and different individuals may choose different paths. Not every individual wants to adapt in ways that society suggests they should. And I know these are important conversations in families.
Of course, yeah, everything is so nuanced and individualized in those conversations. We did have a question kind of about current events, and I'm curious about your take on it. The question is, our son has taken part in many autism studies. Do you think it is important that we take part in the new registry that RFK is putting out? I'm not comfortable with where this information will go, and what's your take? And before you answer, doctor, I'd like to give a little bit of context here too, where we've been following this as an organization. Just it was very concerning, right, when we first heard about it.
Sure.
And from what we've seen, RFK and the Trump administration has kind of now stepped back and said, "Oh, wait, we're not going to make a new list. We're going to use, existing information to kind of compile this research." Is that what you're seeing, and how concerned should families be?
Well, there are several questions there. Should
Yeah, sorry about
your
that.
family participate? What do we think about the information that's being collected? There is a lot. There has been a coalition of autism scientists. We put out and signed on to a letter indicating that a great deal of research has been done over the last 30 years on autism. We've learned a great deal. And as you've heard from Dr. Manzini and Dr. Halladay, we know a great deal about genes and environment, and now genes and environment are being studied together. So we know a fair amount, and we also know about treatments. On the other hand, given different genetic predispositions, there may be specific gene-specific or pathway-specific treatments that are emerging based on everything we've learned. I am glad to hear that the Department of Health has been hearing the population pushback, because I think it's not clear. I think one way to think about it is when you design a study that deals with thousands of individuals and you ask about data that may have been acquired for other purposes, you don't know the quality of that data. As a clinician who has to use electronic medical records, I know that the information I get comes from an individual who's making a report. Parent report or a child report is only as reliable as it is. So there's great deals of variation. And as Dr. Halladay said yesterday, when we have lots of information about associations, this and that both go in the same direction or they go in opposite directions, that's an association that's then a question. Do those two things that travel together mean they're causal? Or are they both due to some other thing that's not involved? And so I think there were concerns about which data sets. As we keep in mind, our data sets that were with the government are supposed to be private and regulated and used only for the purpose for which they were acquired. So opening up data sets that were acquired under one set of rules and using them in a different way doesn't seem to be fair. As a scientific experiment, it's really important that data is collected. Usually the best way is prospectively, where we know what we're thinking the variables are, and we have the right size of the population, and the tests that are done statistically are valid. And so there are excellent tools for doing human population studies for environmental factors, and those decades of methods that are reliable and reproducible using validated measures, not simply somebody says yes or no, those would be much more informative. So since there's not that much money for autism research, I think it would be better for us to continue doing the kinds of research that we've been doing. And for all the families here who are interested in autism, keep in mind that there's 20-fold more money being spent on Alzheimer's disease research than autism. Yet the numbers of individuals in the US with Alzheimer's and autism are about the same, about 6 to 7 million. And so we might want to talk about increasing funding for neurodevelopmental disorder research.
Great. Thank you, Doctor. This has been very enlightening and helpful. And we're going to wrap up here, but I did want to thank everyone once again. We've absolutely loved having you guys, and we really hope to see you again in some other format, whether it's another webinar or we meet you on social media or at our annual conference. Autism New Jersey has a lot of services and a lot of ways to connect with you and help you connect with each other. So feel free to give us a call again, 800-4-AUTISM. And with that, we'll say thank you and goodbye for now.
Goodbye.